Hi, my name is Shawana Piggy, and I am an MSHS student in laboratory medicine in the Department of Laboratory Sciences at the George Washington University. This course is Advanced Molecular Diagnostics, and the professors in this course are Dr. Marcia Fermani and Dr. Jude Aberdeen. In this lecture, we will be talking about the molecular diagnostics technique of fluorescence in situ hybridization, also called FISH. So you're wondering, what does FISH stand for? So let's start with the F. The F in FISH means fluorescence, and a fluorescence microscope is used instead of a light microscope, which is used in karyotyping. And this allows to determine the characteristics of a gene, whether there's a mutation or a deletion through the use of the fluorescent image. And this allows for more dynamic analysis of the specific gene or genes of interest. And to the right, you can see the fluorescence microscope and the computer, which helps to visualize the images. What does the IS mean in FISH? The IS stands for in situ. According to the Molecular Diagnostics textbook, in situ means in the cell. According to the diagnostics textbook, it also describes that this method can detect the DNA, RNA, and basic protein structures within the cell. This end result allows for the visualization of these structures at the cellular level. Lastly, what does the H mean in FISH? The H stands for hybridization. In this method, probes are used to hybridize to specific sections of DNA, RNA, or protein. These probes are also fluorescently labeled. In the image to the right, you can see a fluorescently labeled FISH DNA probe which hybridizes to the specific section of the DNA that's being analyzed. These fluorescently labeled probes are then visualized under the microscope, and this allows for the visualization of the specific cellular structures. There are two major types of FISH testing. The first is called interphase FISH testing, in which the cell is analyzed in the interphase. This method does not require any culturing of the cells. The probes are designed in order to detect any mutations in the cells, which include deletions or amplifications. The probes are added to the fixed cells, which hybridize to the complementary strand of the gene, and these signals can detect if there are deletions or multiploidy in the genes. Different colored probes can assist in detecting if there are any translocations in the genes. The second major type of FISH testing is called metaphase FISH testing. In this type of FISH testing, culturing is required for 72 hours. According to the Molecular Diagnostics textbook, cosamide is added in order to arrest the dividing cells in metaphase. The cells are then fixed to a slide, and 70% acetic acid allows for the condensed chromosomes to spread out. Fluorescent probes, which are multicolor, are then added to the slide in order to be analyzed. Telomeric and centromeric probes are used in order for full chromosomal analysis. This type of FISH can be used to assess deletions, translocations, and insertions within the cell. What are some common specimens that are collected for FISH testing? According to Integrated Oncology, which is a part of LabCorp, bone marrow can be collected in sodium heparin or an EDTA tube, and CSF can also be collected in a sterile tube, and this can be analyzed for genes in cancer patients. According to LabCorp, urine can also be collected in a sterile container in order to assess genes in patients with bladder cancer. Whole blood can also be used, which is usually collected in an EDTA tube that can assess microbes that are affecting human cells, according to a research article that describes the FISH method in Diagnostics. This article also describes that these genes can also be assessed in tissue biopsies. The fluorescence FISH images can be used in clinical diagnosis of a patient. In a recent research article, it describes the interphase and metaphase FISH analysis in gene translocations through the use of dual fusion probes. And these probes are multicolored. In image C, you can see the image on the left is an interphase FISH image, and the image on the right is a metaphase FISH image. And in both images, you can see a translocation abnormality involving the BCR and the ABL1 genes. There are many advantages and disadvantages in FISH analysis. Some advantages of FISH analysis include that a higher resolution can be obtained than in karyotyping because FISH analysis uses fluorescence microscope, where karyotyping uses a light microscope. FISH analysis is useful in detecting mutations and deletions within the cell. And according to a research article, FISH can also be used to detect infectious microbes, which include parasites like malaria in the human cells. According to the Molecular Diagnostics textbook, some disadvantages of FISH analysis includes that it is limited to the specific complementary regions of the FISH probe. Photobleaching can occur when the slides are exposed to prolonged light, so it's very important in this method to protect the slides from light. And lastly, FISH analysis is only able to detect mutations where the probes are bound. The references used to create this presentation include the Molecular Diagnostics textbook, a research article describing the genetic diagnostic and research applications used in FISH, and the reference laboratories Integrated Oncology and LabCorp in order to describe the type of collection and specimens used in FISH gene analysis. Thank you for taking the time to listen to this presentation on the molecular diagnostics technique of FISH analysis. Please let me know if you have any questions about FISH analysis.